I'll talk to you about the European Genome-phenome Archive, the EGA, EBI. I gave an introduction to the EGA back in, I think, March or February of this year. So we'll start only with a brief introduction, and then I'll take you into the update to the EGA, which centered around a new system for applying for access to data, which is the Resource Entitlement Management System, or REMS, a new method for distributing data, which is a streaming download client. I'll talk to you about integration with other EBI services, mostly the Other Ebi Variation Archives, and that's the European Variation Archive, EVA, and the Database of Genomics Variant Archives, the DGVA. Then we'll finish on collaborations, and this centers around our work with the beacon, the EGA beacon that we have, and that's in collaboration with GA4GH and Elixir. So EGA is a secure database for the sharing of controlled access data. So what do we mean by controlled access? So these data sets are all free in terms of monetary value to share, but the sample, when collected, the consent form says that it must be contained within controlled access. So sometimes you can't give it out to commercial enterprises, sometimes the sample data is not allowed to leave countries or continents. So what we do is hold data sets at EGA that are controlled by data access committees, and you apply to the data access committee to have access to the sample level data. So we have around 800 studies at EGA that covers almost 250,000 samples, and I'll go through the life cycle of an EGA data request. So a user identifies a data set on our website or via one of the repositories where our metadata is held, so BD2K or GA4GH, the EBI web search, and they apply for direct access to the data access committee. The data access committee reviews the application to ensure that they have the correct metadata, so that's name, affiliation, email address, but also what the user intends to do with the data and what is the topic of the study. And once the data access committee has agreed to give the user data access, this comes back to the EGA where we generate a user account with the correct permissions. So if we have a different view of the studies that we have at EGA, this is them split by technology in the colors and topic of investigation by the trait names on the left-hand side. And we can see by the light blue bars and the red bars that the majority of data at EGA is either exome sequencing data or whole genome sequencing data. And the bottom half of this graph is all about cancer data, so you can see by far and away the largest topic of study and investigation at EGA studies is cancer. So when you're within the EGA website, this is what a study page looks like. You can see at the top that we have the name of the study, a description of the study, which gives you a brief highlight of what's been investigated. Then we also have the name of the data providers and the data sets. Now, one study can contain one or more data sets. And when we look at the data set page, it's at the data set level that EGA grants access. And it gives you here some information about who you have to contact in order to access these data, and some metadata about the individual data set. So this is a figure which explains what I was saying about how the Data Access Committee controls access. So you have the data housed at EBI, and you have the user that wants to access the data, and they do so by applying to the DAC. And they have to go through the DAC in order to access data. So it's important to stress that we at EGA play no role in the decision making. That's solely down to the DAC, whether a user is allowed to access a data set or not. And once the account has been agreed upon, EGA generates that account. Now, with the current methodology of applying through the DAC, it's difficult for EGA to trace how many applications each DAC is receiving, or how long DACs are taking to reply to the users. Also, we don't really know if the DACs are keeping records of the users that they grant access, and whether these records are being kept consistently, whether they're in databases, what types of databases, what's the schema. or simply on spreadsheets. We don't know. So this takes us to the next part of the presentation, which is one of our most recent updates, which is the use of the REMS system for applying for access to data sets. So REMS, as I said, stands for Resource Entitlement Management System, and this provides a transparent interface between the application and the participating DAC. And this is an Elixir-funded project, and the full application procedure is taken out right within the REMS infrastructure. So this is what the REMS page looks like. On the left-hand side here, we have a typical application form. Now, as I said, the DACs can have idiosyncratic rules that you must adhere to in order to apply for access to data. So some of these questions might be different depending on what DAC you've applied for access, but it takes general information like research project name or the study topic that you're investigating. Now, REMS can store these answers in a local database so that if you apply to more than one DAC and the questions are repeated, you can simply use the previous information. From the EGA side, what we're able to do is monitor applications in REMS. So this page shows that we have 4 outstanding applications. You can see the name and the title of the application. We can also view the application form. We can see when it started, so these have been hanging around for a while. And if we choose to look into any individual application, or if a user comes back to us and says they've been waiting for a long time for a decision, then we can look at the log of the application to say, yeah, okay, Jeff has applied, but around about February, he asked for an update, and this still hasn't been actioned. So this is when we at EGA can step in and contact the DAC directly and see if we can chase this up. From the DAC side, this is a lot easier than dealing with individual application forms because the answers are already stored in a database and they can be automatically emailed at weekly times, and they can just set aside maybe 1 or 2 time slots a week to approve access to EGA accounts. And again, once the EGA account is generated, it comes back to EGA, and that's automatically emailed to the user so that they can access the data. So once they access the data, this brings us to the second update that we have at EGA, which is a new distribution service. So this is a new HTTPS-based distribution streaming service, and it's based on the microservices architecture. So it's a Java client that you can use either at the EGA website via the GUI, or you can download and use it at the command line, and it permits parallel file downloading and automatic restart. So if your internet connection is broken for any reason, or if any file fails, then it just retries that file for up to 100 times. It ensures the validity of the transfer by using MD5 sum checks. So this is all done behind the scenes so that the user doesn't have to worry about it. This slide hasn't really come across too well, but the objective here is that it explains the EGA download use case. So on the left-hand side, we have the EGA database, where we have the EGA data encrypted in our vault, and that's, we call it CleverSafe, or the software is called CleverSafe. And on the right-hand side here, we have a user who essentially wants to access this data over the internet. So this is how we do it. We take the data, the encrypted data from the EBI EGA vault, decrypt it with the key that we have at EGA, and then re-encrypt that data with a password that's set by the user. So when the user uses the new streaming client, they provide a password which they can use to decrypt the data at their end. We have this streaming client in production. We're in version 1.1.5. This is the website that you can read more about the client. It has the help documentation. It explains what the client does. And if we look at the statistics, then it's really a marked improvement on how fast we were able to share data previously. So in testing, we can get about 26 terabytes shared per day. And if we look through the month that this system's been in production, then we started off with about 22 terabytes, and that's when we were going out and telling users, you know, we have this new service, would you like to try it? And last month we reached almost 300 terabytes of data shared using the HTTPS streaming downloading. If we look at who's applying for access to these data sets, or who we're sharing these data sets with via the client, then 33% goes to the US. There's a variety of European countries that make up about another third roughly. We have a huge chunk in Japan, as you may expect. A lot of it's in the UK, 13%. And that covers basically what I want to say about the updates to the EGA. So the next section is about integration with other EBI services. So I'll talk about the European Variation Archive, the EVA, and the Database of Genomics Variants Archive, the DGVA. The split between these archives is that EVA takes short genetic variations, so that's less than 50 base pairs, and DGVA takes structural variations, or classically those that are more than 50 base pairs. So EVA is represented by this logo on the left-hand side, and that should show that we accept all types of variation up to 50 base pairs that can be germline, somatic, within populations, between strains, and between species even. But importantly, because we are tied to the VCF format, basically EVA is a collection of VCF files, then any variation within EVA is represented as a change against a reference genome sequence. When you go to EVA, the website at EBI, this is the study browser, which shows all the data sets we have. I've used the human filter here on the left-hand side. That's most relevant to this call. And we can see a number of data sets. Now, we're trying to integrate EGA data with EVA. So the important distinction is that EVA is completely open access. If you follow any of these links, you can download the VCF file. So when we're considering EGA data and EVA, it's the aggregate level data. So what I mean by that, so you have your, we'll use an example. So we have an example here of the UK 10K data. So the UK 10K data is held at EGA. It's 10,000 genomes, a variety of VCF files with genotypes. And the sample agreement forms say that this data must maintain controlled access. So that's fine, it's all at EGA. Now, what we can do is generate an aggregate VCF file from the data that's in EGA, and that just stores all the variants across all the samples and puts it into one file without genotypes, without annotation. And we put that file into EVA, and that's the file that you can see here. So the point of this is that applying for access to data to EGA, even with REMS, can take a bit of time, and also you're downloading rather a lot of data at a time. So with this tool here, which is called the EVA variant browser, you can use the filtering options on the left-hand side. So we're looking at the human genome, we're looking at a chromosomal location. You can look for certain consequence types or certain protein substitution scores, and you can look in certain studies. So if we look at the UK 10K data, then you can find the variants that are only in that study. You can see if these variants are interesting to you, and then you can choose whether you want to apply for access to EGA for the complete data access. So my call's gone a bit fuzzy here. Are you guys still there?
Yes, we can.
Hello?
Yes, we're still there.
Okay, good. So once we see the data in the variant archive, we can also look at the VCF files via the IOBio suite of tools. So this allows you to look at any VCF file and look at things like variant density or allele frequency spectrum or the base changes that you see. You can look at variant quality, and effectively what this does is allow you to judge the quality of a data set, and then whether you want to download that data set or whether you want to apply for access to the raw data at EGA. We also have a complete API for EVA. Effectively, the variant browser that I just showed was our graphical interpretation of our API. So anyone's free to use the API and plug it into their framework or generate any views that they want of the data. The second archive is the Database of Genomics Variants Archive, DGVA, for structural variations, those that are more than 50 base pairs. This is the front end when you go to the website EBI. And this is the data download page, which gives you the GVF files for these variants. We have about 200 studies. They're all called the name of the first author and the year that they were generated. So these hyperlinks take you to the paper. And the reason I'm explaining these archives is now that we have EVA in our suite of resources, we have archive relationships with those at NCBI. So the relationships here are EGA for controlled access with dbGaP, EVA open access with dbSNP, DGVA for structural variations for dbVar, and clinical studies that come to EBI are shared with ClinVar. And really at EBI, we're pushing the integration of data sets across EGA, EVA, and DGVA. So whether you come with open access data that can go into either of these two archives, we ask you for the core data to see if that can go into EGA. And if your data is in EGA, we ask you to make aggregate files so that this can be shared via EVA or DGVA. And the benefit there is that the open access archives can push the variant data to genome browsers, things like Ensemble. We can also push the data to dbSNP and dbVar so that the variants themselves are given steady accessions. So we're going now to talk about collaboration, and this will center on our work with GA4GH and Elixir, and it's around about the Beacon project. So GA4GH, I'm sure many of you are aware, Global Alliance for Health and Genomics and Health, and it's a global project or consortium about data sharing. We are part of a number of data work, GA4GH working groups. This one's the data working group led by David Hasler and Richard Durbin. And our involvement here is to generate a beacon. The Elixir project is an infrastructure for Europe's life science research. This is a map mostly of Europe, where we can see EBI is represented here. And the point of Elixir is that there are a number of nodes in European countries that are linked together based on this framework, and each of the nodes builds something that's based on their national strengths and priorities. So in EBI here, unsurprisingly, we have bioinformatics and data infrastructure. So these collaborations are focused on this Beacon project I mentioned. So the Beacon project is basically a first pass project which tests the willingness of collaborators to share data. It's a simple search where you can take a position in the reference human genome and look for the presence of an allele within the data set that holds the beacon. The beacon has 3 tiers. First tier is public. That is fully public, so anyone with the web access can go to a website and put in a position in the genome and say, do you have X base? So a position, 3 million on chromosome 5, do you have a C in your database? And you simply get a yes or no answer. The second tier of the beacon is a registered access. So to access the beacon, you must be registered with the resource that has the beacon. And that's an example here of EGA beacon. We're a registered beacon, so you have to have an EGA account in order to access the EGA beacon. The third beacon tier is controlled access. So that means controlled access. Hello?
Hello.
So the third access, yeah. So the third tier of the beacon is controlled access. So that means that in addition to being registered with the site that has the beacon, you also have to apply for access to the beacon. And at EGA we have examples where if you want to, so because we have a number of data sets hosted by different DACs, if you want to use the beacon to look in all those data sets, you have to apply to a number of different DACs, and that's called controlled access. So this point here says that at EGA we can theoretically be seen as a beacon of beacons, right? So you have a beacon for each data set that we have, which can have its own tiered access. Some of them can be public, some of them can be controlled, and effectively we're a beacon of beacons. So this describes what I've just explained in a graphic. On the green here, you have the public tier, which any anonymous user that's using the web can come and check your beacon. In the orange, we have the registered user, so you have to be registered at the site that holds the beacon, then you can check it. In the red, we have the controlled access. So in order to use this beacon, you must apply for access to a third party, which then says you can use the beacon to see the data. This is what the EBI or the EGA beacon looks like. And this is in collaboration with our EGA partners, which are CRG in Barcelona. So you're looking at the reference genome, you select a data set, you select a chromosome, you select a position, and you look for any allele. And as long as you have access to at least one data set at EGA, you can use the beacon, and the result is given to you either as a graphical interface, or we also give you a JSON. So I'll conclude there, and I'll talk to you once again about the EGA, which is the Controlled Access Archive at EBI. One of our updates is REMS, the Resource Entitlement Management System, which is a transparent application process. We have a new streaming download client, which gives up to 26 terabytes a day. I've also spoken about the EVA and the DGVA, which we're integrating with EGA data, and I've spoken about the collaboration on the Beacon project. So I'll give my thanks to the following people. Justin leads the group. The rest of the guys work on EGA at EBI. Our partners with EGA are CRG, and these are the guys that work there. And we also give our thanks to Ensemble Variation and the guys that work at EVA at EBI. So I'll stop there and take any questions. Thank you.
Okay, thank you, Gary. Are there any questions?
This is Andrew here. I have a question. So the streaming client is about getting data into the hands of people. So that sounds fantastic. I'm wondering if you've thought about other sort of client-side libraries for directly importing that into sort of compute environments. I'm thinking like Python clients or R clients, or in the context of our BDQ Center, even something that brings data into the Synapse infrastructure.
Yes, we have. And we have a Python client which is being generated, and that uses the user side. But at the moment, really, these things are driven by the help desk, and all the users that are coming to the help desk have been able to use the streaming client in order to access the data. So there really isn't much need for us to focus on anything else just yet. For the 220 institutes that we serve data to, only 2 of them have been unable to use the HTTPS client, and that was because the firewall of the institute was so severe. So, so we've got a good record with this client, and if we start to find the use case that people can't access the data, then we'll start to look at our Python client some more. But at the moment, it's really background work for us.
This is Robin Park speaking. And hello?
Hi. Yeah, we can hear you.
Can you hear me? Oh, okay, you can hear me. Okay. Your last slide showed that the daily users download more than 26 terabytes. Is it correct?
Yes. Perhaps I said that wrong. So what I'm saying is that 26 terabytes per day was to one user. So only one person was. So one individual account is able to access up to 26 terabytes per day. But in terms of
it means the people, whatever one user downloaded more than 26 terabytes per day.
No, 26 terabytes per day is the most we've been able to do. But it's getting faster, and your engineer thinks that we can do a bit better as well.
Oh.
Okay, are there any other questions? Okay, thank you, Gary, for that presentation. So next we have Robin Park. So our second presentation will be presented by Robin Park from Scripps. Today he will be presenting about the integrated proteomics pipeline, which is a proteomics data analysis platform. Without further ado, allow me to introduce Robin.
Hi, yeah, this is Robin Park speaking. So we have the IB2. This is just, I think, the second presentation at B2K, and we have the new version. Recently, we released the version 3, and we started integrating many different the tools into IB2, and also we started collaborating with the different groups, research groups, and we have IB2 features. They are only the main features that we already have. Spectrum quality control, and also the user can track down protein database, and also they can easily manage a large number of proteomics protein databases. User can also easily organize large proteomics data through the IB2. Also, people can share the project with the collaborators. We have different protein database search engines like Sequest, Comet, Prolucid. Also, we have a big database search engine tool, and also recently we finished developing a blind PTM search engine. We also provide a comprehensive quantitative analysis tool by using Census version 2. We also provide many different statistical tools, and also PTM analysis tools, and also we provide gene ontology tools at functional analysis page. User can also generate a large number of spectrum annotated image for publications. So then what you do in IB2 version 3, we completely redesigned the interface, user interface, and also we have a new intensity-based label-free analysis tool. And also user used to download the Census GUI to view the reconstructed chromatogram, but now everything is embedded, so you don't have to install any additional software. And also we integrated Metabolomics tools, and also user can convert raw data automatically in IB2, so they don't have to run another standalone software like a raw converter or a raw extractor to generate spectrum file. We have a gene ontology tool, PSA Quant, and Ontologizer, blind PTM tool, and also user can submit batch search, nude TMT tool, and also we recently started collaborating with the de novo group. Then we integrated the de novo search engine tool into IB2. So this is the main login page of IB2. And after login, user can see that this is the project page, and the overall structure is pretty similar to previous one, but our interface has been changed. And let me see. So this is one of the example that quantity result page. So user used to click and launch web start to start looking at spectrum viewer, but it require Java software on your desktop, and somehow the recent the Java virtual machine, somehow it started the security the level become more like become higher, so user have to tweak to launch properly on in window control panel. So it's doable, but it can be usually often can get confused. So this is all embedded. So this is a protein page when I select the protein, and this is a triple dimethyl labeling. So they are peptide for this protein, and when I click select some of peptide, user can see the whole. Oops, let me see. Oh, sorry. So user can. So when I click the peptide, user can see or peptide on the right side, and people can see different, the reconstructed chromatogram, so they don't have to install any software there. And search engine, we have again several different search engines. One of our major search engine is Proteomics, and we recently published a paper at Journal of Proteomics. And this is one of a table from the paper. So we compare the different search engines, Sequest, Comet, and Proteomics. And on here, we use the HeLa and like a fully triptych search. We can get more ID comparing to other search engine. I compared actually the more search engine other than the Sequest and Comet. Proteomics always get actually more IDs. And the next table is a half a triptych search. This is more like a more common search. So we can see even bigger gap comparing to Comet. Comet is its algorithm is based on Sequest, so you can see Comet and Sequest, they are pretty similar, pretty comparable. And next one is gene ontology. So gene ontology, we have 3 different categories: biological process, molecular function, cellular component. Traditionally, we calculate p-value based on gene geo-enrichment. And for the the conventional the gene ontology analysis, we are using Ontologizer. But also we have a new tool, PSA Quant, in which is developed at Johnny H. Lab, Matthew. This tool also integrated into IP2. So this, the concept of a PSA Quant is different from a conventional approach, because we know that a group of genes can co-express. Conventional approach, we assume independent, all the genes express independently, but biologically it's not really true. So PSA Quant, it actually grouped the gene and then analyze it. And also PSA Quant, another big advantage of PSA Quant is that it can take the quantitative information as input, like a spec count or the protein ratio, and then it analyzes it. So one of Quant page or the ID step page, you can click PSA Quant, and this is the input page for the PSA Quant. The screen is a little chopped off. But there, if you scroll down in the main actual IP2 page, you can see there are a whole bunch of parameters, and then at the bottom you can click submit. And this is the Ontologizer page. Still, you can use the both Ontologizer and also PSA Quant, and you can select the organism, and then you can specify some parameter, and then you can run it. And this is a geo terms in pie chart, and also there is all the p-value and ratio. We have a new tool, Census version 2, for label-free analysis. We have spent more than 6 months, almost 1 year, to improve the label-free analysis algorithm. So this is an interface for label-free analysis. So last ASMS, Dow Chemical, they presented by comparing Census 2 through the IP2 and Skyline and some other tool for label-free, and it show the better result through the by the Census and also combined with IP2. I don't have the result or the comparison table, but you can find it from ASMS, the website or their resource. Let me see. So this is a label-free page. This is a fusion data. So this is again the protein information. When I click, I can see all different peptide, and I can select. And we have 3 different experiments in this case, and we can see that same peptide elute it through at a different retention time, but they're slightly different. They're pretty close. And so in this case, we have only 3 experiments, but in reality, usually can have more commonly like a 3 control, like a biological or technical replicate, and also some biological sample, again, like a triplicate, and then they can compare to calculate all different p-value and some other score. Another one is Metabolomics. We at the H Lab, we also have work on Metabolomics tool. So the current, the limitation for the Metabolomics data analysis is that when there's overlapping peaks, they are not properly analyze the overlapped peaks. Unlike the proteomics, metabolite, there are many different metabolites. It's very common that there are the mass of metabolites are pretty similar, and also but the chemical structure are slightly different, in which means its mass can be very close, and also its retention time also they can elute in a very close retention times. So the multiple peaks can appear elute very closely, so it's very hard to differentiate those metabolites. And also there is no proper scoring function to differentiate the good metabolite signal and also the bad one, and also it's just slow to analyze it. So we have a new tool called Dilute, in which is developed by Tom at the H Lab. So this is one example, and this is a pretty common, the issue at Metabolomics. So this is the overlapping peaks, and we obviously know that there are 2 metabolites co-elute. So the conventional approach, existing tools, they just cut in between these 2 peaks and then calculate the area. But this is not really correct. So our approach is that we model these peaks, and then we separate them, and then we calculate the peak area under the curve, in which can generate a more accurate result. So there are actually a couple of the steps to detect such peaks. The first, we reconstruct the chromatogram, and then we fit into a statistical model, and then we calculate all score for each peaks, and then we compare those peaks. So this also integrated into IP2. So we have, in this case, we have 6 different experiments, and then there are all different mass that are automatically extracted by software. And when I select, we can see that there are 6 different Metabolomics experiments, and the light color on the graph is the actual data, and then dark blue, like a solid blue, that's the one that we fit into a statistical model. And we can see all different the metabolites. And again, in this case, the first 3 the control, and then the other 3 biological sample, and then we calculate all different score for that. Another tool we integrated into IP2 is de novo search engine. So we collaborate with the Bigma group in Canada, and this is the IP2 page. And this, the novel was presented at last ASMS, so this year. And there you can see that there's a novel search in the option, so user can select, and then there's some input parameter, and then user can submit. So this, the novel, compared to other de novo search engine tool, user claimed that it can generate a more accurate result, and also when we test it, it's just super fast. So you don't even need any cluster or other additional computational resource. We just run on on a single server, which is good enough. So next one is blind PTM search engine. I think this has been presented, I think, several weeks ago by Lin. So this is the blind PTM search engine tool, in which is complete, and we are actually working on integrating into our pipeline. So its idea is that we have a protein database, and this is typically the search engine. So like Prolocid or Comet or Sequest, and we get a search engine, and then we find our protein, and then we build the small database based on based on search engine search results, and then we search in blind PTM tool, and then we can combine the search engine and then blind PTM result, and then we present the final result. So this blind PTM, again, as you know, the biggest advantage is that you don't have to specify PTM. So you can automatically check and try to find all possible PTM. So it's a very useful tool, especially when you don't know what kind of PTM happen in your sample. Another one is the raw to MS2 file conversion tool. So people used to run raw to MS, used to convert the raw file into spectrum file by standalone software like a raw extract or raw converter. But the raw to MS2, this tool has been developed by Tom at EH Lab. So you can simply upload a raw file. Then there's an option on the web page that you can just check the conversion mark, and then it will generate automatically behind. And another one that we are working on is that data integration. So we are going to allow user to click and then integrate the proteomics data into different like a repository like Pride or the Massive. We try to automate the data integration into the Pride, but there's some the technical question. Hopefully the next week we have a meeting at UCLA, so hopefully that we can discuss more. I can bring up some questions for EBI group, and so that's the next one that we are working on. Okay, I think that's pretty much.
Thank you.
Hello.
Yes, thank you, Ron. Are there any questions?
Robin, this is Andrew here. I'm interested in this blind PTM search. Can you talk about what you've done as far as analysis or validation, or where that's going in the near future?
Okay, the tool has been done. Where is it? Okay, the tool has been developed. So what I'm working on is that this the individual, like the search engine result, and then the generating the small the database file, and also the running blind PTM search like BLASMAS, BPS. So this individual one, we have a small script, to run that. The major one is, of course, the blind PTM search engine. So this part is done, but I'm trying to automate, combine all those small script so that it should be easy to use. And also another thing that I'm working on is to generate, job file automatically. So like, the Prorucid or Sequest for now, the user can run, the Perl script that we wrote, or, and the people can just type, and then it will automatically split and then run in cluster, split file, and then distribute to the node in the cluster. And these, things can be very hassle, but that's the one that, but writing script is pretty easy for me. So I'm just trying to integrate. So right now, all tools, individual, the step, they are done. Just, we like to, put together. And that's the next one. And validation-wise, the detail select is going to actually put, all together and then calculate, the FDR. So that validation is pretty, the standard, the validation that in the proteomics that we are using it. So yeah, I think that should be, as long as the user can define the proper FDR, I think that should be good enough.
And do you have immediate plans to apply this? I mean, we've been thinking about this very superficially recently, and one of the and of course we're learning that it's a hard problem, right? As you get lots of different combinations now. So anyway, I'm just wondering, in terms of biological applications. Or even, mining existing data sets for novel PTMs. I mean, any thoughts there?
Well, so this, the blind PTM that I'm not sure if I understand your question correctly, but this blind PTM tools. This approach is pretty well known. Only the issue is that how we can develop, the good, and fast, the blind PTM search tool. At the HLab, we already developed the blind PTM search tool before by Tao Xu. We just tried to, upgrade, make, the better tool, as we know that there's some drawback in the some limitation in the previous tool. Based on our experience, we actually developed a better tool. That's the BLASMAS BPS. So definitely this will be very useful tool. And so even though I didn't package all of them yet, but that they should be quick. Probably in a week, I think I can make ready for people to start using it. Even though it's not I packaged it yet, but already the last the many weeks I already got the request from people that they are asking me that it's ready. So I think definitely this will be very useful tool. The limitation for the blind PTM in the past was that it's slow. Because this is kind of the exhausting the search, but now we have pretty fast the algorithm, and also we have good strategy. So for example, this instead of using standard, we can actually split the spectral file into much smaller, and also better algorithm, and also the more automated way. Definitely we can make a much this all the whole analysis much faster. So yeah, hopefully I think it will be ready in a week also. So I also talked to one of your lab, I think postdoc or graduate student, I think a couple of weeks ago. So definitely I'm going to push it to make it ready, and then we can actually discuss further.
Great, thanks, Robin. Sounds really interesting.
Yeah.
Okay, are there any other questions for Robin? Okay, well, thank you, Robin, for that great presentation. I'd like to remind everyone that we will be taking a 4-week break from the conferences starting next week, August 21st, until the 11th of September, and we will resume our conferences on September 18th. So feel free to send me an email if you have any questions.